I want to share with you today some of our latest progress on research that we're doing for aluminum tolerance using both genome-wide association and QTL mapping in rice. First, why rice? Rice is one of the most important food crops in the world. It provides about 20% of global human calories. Rice is the staple food of Asia, where 90% of the world of the rice production in the world is consumed, and also where most of the half of the world's poorest population lives. And although we tend to associate rice primarily with Asian culture, rice is also deeply embedded in Latin American culture, where I come from. And as a staple food, rice is becoming increasingly important both in Latin America and in Africa. And it should come as no surprise to those in this room that the global demand for rice is predicted to increase by at least 26% in the next quarter of a century, both because of incomes rising and population growth. Rice, genetically, is as diverse as the cultures that consume it. And this diversity is organized in a particular way. And this has implications for those working in rice genetics. Rice has a very deep subpopulation structure, as illustrated by this phylogenetic tree that was built using 170 SSR markers in a subset of our rice diversity panel of about 200 rice varieties. And rice is subdivided in two subspecies or varietal groups, the indicas shown here in red and the japonicas shown here in blue. And these varietal groups can be further subdivided in 5 subpopulations: the tropical, temperate, and aromatics that constitute the japonicas, and the indica and the aush that constitute the indica varietal group. So this deep subpopulation structure means that allele frequencies will vary greatly both within and among each one of these groups. And the way this diversity is partitioned will determine how and whether it can be detected. So this has implications for genetic mapping. And in the case of GWAS, one can and should do GWAS in a couple of different ways. You should use your entire diversity panel, considered of all different subpopulations. And in order to detect alleles that are varying in frequency within your subgroups, you can also run it within each varietal group, as well as each independent subpopulation. So also, GWAS will work well in combination with biparental QTL mapping, particularly in rice, where it's common to see crosses between parents that are coming across different subpopulations or across the subspecies, indica and japonica, as a way to unravel variation that is confounded with subpopulation. So we're working on rice aluminum tolerance, mostly. I'm sorry, I'm going to move over here because I can't see my slides. Sorry, OK. So we're working in rice because not only rice is an important food crop, but it's also a very resource-rich model species. And interestingly, rice is much more aluminum tolerant than most crops, and it's by far, by several-fold, the most aluminum tolerant cereal. So the objective of our project is to understand genetic variation in key aluminum tolerant loci and how it contributes to phenotypic diversity in rice. And we hope to use this as a foundation for crop improvement, not only in rice, but also in other cereals. So this is a brief outline of my talk today. I want to give you an introduction on the problem of aluminum tolerance and also a problem of aluminum toxicity and a background on aluminum tolerance. And then I'm going to tell you 2 stories. I'm going to tell the story of a gene that we detect via genome-wide association, and then of a gene that we don't detect via genome-wide association, but we detect via QTL mapping. And I want to stress the point that a lot of this work that I'm going to show you today is work in progress. And so you're not going to be a big conclusion slide at the end, but I think it's more interesting to show you real-time data and also have a chance to get feedback from the department. And I also want to mention that it's largely a group effort, so I'm going to do my best to try to thank everyone involved that contributes to the project as we go along. So first, the problem of aluminum toxicity. Aluminum is the 3rd most abundant element in the Earth's crust. When soils are neutral, aluminum is bound to other elements and it's inert. However, when soil pH drops below 5, Al3+ is solubilized. It's toxic to the plants because it can be absorbed by the roots. So what this map is showing you here is the distribution of aluminum toxicity expressed as a percentage of aluminum saturation. And you can see that there are vast areas of the world, shown here in blue, with high saturation of aluminum in the soil, These overlap with arable lands in the world and are concentrated in the subtropics and the tropics and in regions where food security is most tenuous. AL3+ inhibits root growth and function, and the stunted roots also leave plants more vulnerable to drought and mineral deficiencies. And these are some classic illustrations, both from maize and wheat. This is a photo taken by Embrapa maize and sorghum showing aluminum-resistant and sensitive varieties grown in the acid soils of the Brazilian Cerrado without any correction. And you can see the stunted growth of the aluminum-sensitive variety. And these are wheat seedlings grown in acid soils, and you can see the stunted roots of the aluminum-sensitive variety, and an electron micrograph that shows the damage that aluminum does to the meristem of the growing root tip. Plant physiologists have been working for many years in understanding the mechanisms of aluminum tolerance, and this is a nice illustration from a recent review that covers all different mechanisms. And I would like to stress the point that the aluminum exclusion mechanisms is by far the most widely recognized and adopted by a variety of different plant species. And the way this mechanism works is upon perceiving the aluminum stress, the plants activate transporters at the plasma membrane that release organic acids. These organic acids complex aluminum in the rhizosphere, and this complex cannot be absorbed by the root. In most recent years, some different mechanisms have started to be unraveled in different plant species, and some of them involved actually bringing aluminum into the cell where it's complexed with either phenolic compounds or other unknown compounds, and it can be also sequestered into the vacuole. In recent years, there's growing evidence for the involvement of the cell wall in aluminum toxicity, but the mechanisms of this are by and large still unknown. Finally, there are a couple of species such as buckwheat and hydrangea that are aluminum accumulators, and what these species do is to load aluminum into the xylem, translocate it into the shoots, and then sequester it into the vacuoles. And in case you didn't know, this is what gives hydrangea its beautiful blue color in acid soils. In terms of genetics of aluminum tolerance, several QTL studies have been published in a variety of different species, and it's really by and large a quantitative trait. However, a few alleles of large effect have been identified in different species. And in the case of these large effect QTLs, they have been, in most cases, map-based cloned and shown to be the gene that actually encodes the transporter involved in citrate release or malate release, depending on the species. This has been the case for wheat, maize, sorghum, and barley. Now, when we talk about aluminum toxicity specifically in rice, I think it's really helpful to look at it in the context of the different rice ecosystems production, because they do vary widely. So what you see here is a map of the different rice production ecosystems, where in blue you see paddy rice, which is irrigated rice that's grown under constant flooded conditions. And then in yellow is rain-fed lowland rice that goes through periods of dry or wet, depending on rain. And then finally in red is upland rice that is grown without any flooding. So what's interesting is that it's widely recognized that when rice is grown under flooded conditions, the pH of the system is determined largely by the pH of the water, and it stabilizes in around 5.8, where Al3+ is not toxic. So we believe that there's limited aluminum toxicity in the case of irrigated flooded rice. However, if you look at, again, the map that I just showed you showing the areas of high toxicity of aluminum in the soil, these areas overlap largely with areas of upland and rain-fed lowland rice, and these are concentrated in South America, West and Sub-Saharan Africa, as well as Southeast Asia. This is the platform that we use for studying natural variation in rice and that we are applying for aluminum tolerance. And so we have developed a collection of germplasm in collaboration largely with IRRI and the USDA. We have about 1,500 purified rice varieties that cover all rice subpopulations, and were collected from over 100 different countries in the world. And these varieties have been genotyped using an Affymetrix high-density array with over 700,000 SNPs. And the publication that describes both this platform is currently provisionally accepted in Nature Communications, so we hope to have this out in a few weeks or so. What I'm showing you here is the distribution of polymorphic SNPs in the different subpopulations to to stress the point that even though allele frequencies will vary widely in the different subpopulations, with our high-density array, we do detect a very, very high number of polymorphic SNPs that gives us really high resolution for GWAS within each subpopulation. So in terms of phenotyping, we have developed a phenotyping platform that was developed a few years ago by a couple of grad students that were working in collaboration between Susan's and Leon Koshin's lab up at the USDA, across the street. And it was work done by Adam Famosa and Randy Clark, where they adapted the hydroponic system that was in place in Leon's lab for growing rice, which requires much higher levels of aluminum in the solution in order to see a phenotype. And then developed also a digital imaging platform for accurate measuring of roots. So we phenotype on seedlings in hydroponics. We grow the plants both in control and under aluminum conditions. After a few days, we photograph the plants, and then we use an in-house developed software called Root Reader 2D to extract the total root length. And then we use an index by dividing the aluminum-treated growth by the control growth to control for just natural variation in the rates of root growth. And that's how we develop our aluminum tolerance index phenotype. We have applied this phenotyping platform to a subset of our diversity panel of about 400 varieties, as well as 2 biparental mapping populations to do both GWAS and QTL mapping. And we have done GWAS using our previous SNP phenotyping platform that had 44,000 SNPs. And also we have really fresh from the oven results using our 700,000 SNP array, which I'll show you briefly. And then we have done QTL mapping in 2 populations across indica and tropical japonica and in Ausch versus temperate japonica. And the story I'm going to tell you today focuses on the indica versus tropical japonica population. So this is what the phenotypic variation looks like in our diversity panel. And you can see that there is plenty of phenotypic variation, both within and between the different subpopulations. And it's clear to notice here that the japonicas overall are a lot more aluminum tolerant than the Ausch and the indica. And again, our biparental QTL mapping populations are crosses between tropical japonica and indica and temperate japonica and Ausch. And it's also interesting to notice that temperate japonicas are the population where you have most of the lowland rice that is widely believed that is not grown under aluminum toxicity, and yet they are a really strong source of aluminum tolerance alleles. And don't have a good explanation for that, but it's interesting to note. So I'm showing you here our GWAS results using the 700K array. And as I mentioned earlier, we do GWAS a couple of different ways. So this is what it looks like when you use an entire diversity panel. And this is the indica varietal group, and this is the japonica varietal group. And then when you do it in each individual subpopulation, Ausch, indica, temperate, and tropical japonica. Now we have a few promising peaks that I'm showing you here in purple. However, we are still really working on trying to see what are the candidate genes and do haplotype analysis under these peaks. However, this peak here in red is a peak that was also detected in the 44K array, and we have been working on this for a few years, and that's the story I want to tell you. So under this peak, we found an aluminum tolerance gene that had been cloned independently by a different group, and it's called NRAT1, and this is the location of the gene under the peak. So this gene was cloned by a group in Japan that was working on a rice aluminum tolerance, and what they did was based simply on mutagenesis of a temperate japonica aluminum tolerant line. And they found this gene that encodes a metal transporter that they believe mediate aluminum uptake. So this is an this is a plasma membrane transporter that works in the root tips and brings aluminum into the cell and works in combination with another transporter that sits on the membrane of the vacuole to translocate and sequester aluminum into the vacuole. So following up on our GWAS results came the work of Jonghyun Lee, who's a postdoc in Leon Koshin's lab. And he did functional characterization of the aluminum-sensitive haplotype, which he identified by doing sequencing on a subset of our panel. And what he showed is that there is a specific allele that we called NRAT1-2 that was unique to the aluminum-sensitive Ausch varieties. And what he showed is that not only it had much lower expression of the gene, but it also had lower protein function. So this allele carries 4 amino acid nucleotide substitutions that result in 4 amino acid differences to the wild type that made the protein less efficient in transporting aluminum. So if you look at the distribution of aluminum tolerance throughout different subpopulations, again, these are the lines that carry the 1 and 1-2 allele. So these are all of these carry the what we call wild type allele. Now remember that aluminum tolerance in rice is much more higher than in any other crop species. So we see this as a very basal aluminum tolerance mechanism that is present. It was identified initially only in rice. And so we thought this was an interesting opportunity to do some translational work and see how we can move this into other species. So initially, the first thing Junyan did was to overexpress the gene in Arabidopsis, and what he shows is that when you overexpress NRAT1, the rice and NRAT1 in Arabidopsis, you make Arabidopsis more tolerant to aluminum. As you see here, the plant's grown in aluminum with longer roots compared to the wild-type Columbia. And this is relevant because it means that all the machinery that's necessary for translocating the aluminum into the vacuole has to be conserved between these different species. So it's promising. So moving into crops, the first thing we did is look for actual homologs within each species, and we identified in maize a gene that has makes a protein that has a higher, very high 80% identity at the amino acid level to the rice NRAT. And interestingly, it overlaps with an aluminum tolerance QTL. It's a minor QTL, but in rice and in maize, I'm sorry, this is really highly quantitative. So we think it could be underlying this QTL. And so what Junyan is doing now is the functional characterization of the homolog. And that, I'm sorry about that, slide got messed up. But basically what he's shown is that it has a lot of similarities to the original rice gene. So in terms of gene expression, the gene is expressed in the roots and not in the shoots. And when the plants see aluminum, there is an upregulation of the gene. He also showed that just like the rice, the maize NRAT is localized to the plasma membrane, as shown here by doing infiltration in tobacco cells, overlapping with a plasma membrane marker in red. And then when he overexpressed the maize NRAT in Arabidopsis, he also got a very similar result where he sees that the overexpression lines are a lot more aluminum tolerant than the Columbia. So again, I think this looks very promising. So to quickly summarize what we know about NRAT so far, I think NRAT illustrates really well a case where we can use natural variation to identify key aluminum tolerant loci. NRAT seems to be required to maintain really basic levels of aluminum tolerance in rice. And in terms of translating this to other species, it's possible that maize may have a similar NRAT-based mechanism, and now we're trying to see whether this can also be involved in natural variation in maize. And finally, through engineering, we're trying to see whether NRAT can really increase aluminum tolerance in other species by overexpressing it in wheat and maize. And this is all work in progress. So moving on to the third part of my talk, I want to tell you the story of ART1, which is a gene that we detect in a biparental QTL mapping population. And within this story, I want to tell you about work that we're developing using nearsogenic lines and ongoing work on trying to understand allelic variation for this gene. And then at the very end, tell you what we're moving forward with, which involves genome editing. So this is one of our mapping populations, and it's a cross between an aluminum-tolerant tropical japonica called Azucena and aluminum-sensitive indica, IR64. And so in this QTL mapping, we detected 4 QTLs, and the major QTL here on chromosome 12 explains about 20% of the variation for aluminum tolerance. And when we look closely at this QTL, again, we found a candidate gene that had been characterized by an independent group. This gene is called ART1, and this was characterized actually by the same group in Japan that found NRAT. And what they did was basically take an aluminum-tolerant temperate japonica, mutagenize it using gamma rays, I think, and then look for mutants that were aluminum sensitive. And they found this mutant here that had no phenotype when grown in regular soils, but under acidic soils had a very extreme phenotype of short roots. So they did a positional mapping of this mutation and found that it was sitting on a zinc finger transcription factor, which they called aluminum-resistant transcription factor 1. And this gene being a transcription factor, then they did the obvious experiment. They used microarrays to look at genes that were upregulated when the plants were exposed to aluminum in wild type, but that the mutant was unable to turn on. And they reported on a list of about 30 genes in that microarray. And then what they did was pretty much work their way down the list of genes that were identified and show that they are related to a lot of these mechanisms that I described to you earlier. And RAT is one of them, working on internal detoxification. There is a MATE transporter involved in citrate release, cell modification, and also still many unknown genes of unknown function. And again, this was a gene that was discovered by just induced mutagenesis, and until our GWAS and QTL studies came out, really, there was no indication that any of these genes were involved in natural variation for aluminum tolerance. And ART1 being a transcription factor, then we're interested in seeing what is the effect of this QTL when you work in different genetic backgrounds. So I want to show you now the work that was developed by Juan David Arbeles, who was a graduate student and now is a postdoc here with us in the lab. And he did a couple of very interesting things. He developed a set of reciprocal neurogenic lines where he introgressed the chromosome 12 QTL from the Japonica background into the indica parent, and then vice versa. And he also did the fine mapping of the QTL region. So he used just traditional marker-assisted backcrossing to generate the nils by crossing one of his recombinant inbred lines to the reciprocal parent, and used markers to select both against background and in favor of the introgression. Did several rounds of that until we cleaned the background and come up with a clean nil. And again, he did that both parents, both backgrounds. And once he obtained the lines, he proceeded to phenotyping them to validate the effect of the QTL. So what I'm showing you here is aluminum tolerance for the parents. So this is Azucena showing aluminum tolerance, and IRC64, aluminum sensitive. And then when he phenotyped the nils, what he showed is that the nil with the Azucena background, when you introduce the IR64 region of chromosome 12 into Azucena background, you lose about 30% of tolerance compared to the sister nil that is homozygous Azucena for that region. And conversely, when you introduce Azucena DNA in that region of chromosome 12 into the IR64 background, then you gain about 31% of tolerance when compared to the sister nil that's homozygous IR64, therefore validating the effect of the QTL in the opposite genetic backgrounds. In terms of fine mapping, he was able to bring the QTL down to a small region of only 44 kb that contains 6 gene models, one of them still being ART1, our major candidate. So we're looking at these candidates in the fine map region, and naturally, because of all we know and the molecular characterization of ART1, it continues to be the major candidate for our QTL. But we're also looking at the other genes and looking at their expression pattern, both in silico and running real-time PCR, with hopes that we could see an expression pattern that would help us completely rule them out. And also looking at how the parental alleles of ART1 are different from each other. So what I'm showing you here is the expression pattern using quantitative real-time PCR, and this was developed by Ana Rita Rebelo, a technician in our group. And what she did was measure expression of these genes in the parents, both in roots and shoots, both in control and also in the presence of aluminum. And what we were hoping to find is that some of these genes would simply not be expressed at all in the roots, because that would be really a way to say this cannot be involved in the QTL. However, every single one of them is expressed in the root. On the other hand, none of them really had an expression pattern that made it really, really exciting or make it look like a good candidate, except for Severin 310, which we were surprised to see that has actually a much higher level of expression in Azucena when compared to IRC64. And this is both in the presence and absence of aluminum, as well as upregulation in the shoots of the tolerant line, which was interesting and unusual to see. When we looked at the region of the gene and look more closely, we see that it encodes some type of cell wall enzyme, but it's really poorly annotated and the function is still unknown. But we think this gene requires a bit more careful look. Because of this result, Juan David, together with Tim, who's another postdoc in the lab, decided to go back to the drawing board and do another round of fine mapping. So we currently have in the greenhouse around 20 heterozygous plants from our original F2 population. And we are going to generate about 5,000 progenies and screen them with markers in the region, hoping to find recombinants between ART1 and 7310 that we could phenotype and narrow down the QTL region even further. In the meantime, we are looking at the ART1 parental alleles, and we're looking to see how they differ both in terms of expression pattern and also sequencing them. So what I'm showing you here is quantitative real-time PCR showing the expression of ART1 in our parents and also in the nils, in the roots. So the original paper that described ART1 mentioned that there was not a significant change in expression of the gene in response to aluminum. So the way that ART1 itself is regulated by aluminum is probably at the protein level and still not explained. And but also they didn't have a variety of genotypes to really compare. So we were looking to see whether there was a difference in expression between our two parents. And what we see is that there is not a really strong difference between the tolerant and the sensitive parent. However, we see a trend, a really discrete trend of downregulation of the gene in azucena that is reproducible in our different reps and seems to be statistically significant, although it's a really small and discrete downregulation. Even more interestingly, when we look at the nils, we see that the trends seem to mimic the parents that are the donor of the introgression. So starting to think that this trend might actually be real, but we're adding a lot more biological reps to this to really nail it down and be sure whether this is real or not. In terms of sequencing, we have sequenced the coding region of the two parents, and what I'm showing you here is in a protein alignment where azucena is on the top and RX64 on the bottom. And what we see is that there's a lot of differences between the two proteins. We find 6 single amino acid substitutions, 4 small indels, and 3 large indels in the two proteins. What I'm showing you here is the location of the putative zinc finger domains. These are the domains responsible for the protein binding to DNA. And although none of the differences between the proteins overlap with this domain, it is quite likely that they must have an effect on secondary structure, protein folding, or also affect other domains that we were not able to predict in silico. So our working hypothesis at the moment is that functional polymorphisms that change the ART1 protein could affect its ability to bind to either the promoter or other proteins. So if you picture the transcriptional activation complex sitting on DNA, you have several different transcription factors that interact with each other, as well as with enhancers, activators, repressors, and the RNA polymerase. So you can imagine that a conformational change in any one of these proteins could have a quantitative effect in the expression of the gene downstream. So we're not talking about an on and off switch. This isn't a knockout mutant, but a small modification here could result in a quantitative effect down here. So to test our hypothesis, we're measuring allelic variation in ART1 activity, and we're doing this in a couple of different ways. We can do this directly by just trying to measure protein DNA binding affinity using functional assays, and also indirectly in a genomic context by measuring the expression of the genes that are regulated by ART1. So for functional assays, this is work done by Tim Job, and he started using a simple Wee1 hybrid system. So the literature on ART1 had identified a promoter element of about 165 base pairs that was present in the promoter of the genes that are regulated by ART1. So what he did was to fuse this to a reporter gene and clone it into yeast, and then express each one of our ART1 alleles in yeast and measure the expression of beta-gal, with the idea that difference in this, the binding on the affinity of this binding could have a quantitative effect on the expression of the reporter. These are the results that Tim has so far, and we have measured using both the azucena and the RX64 allele. We're also including some other alleles. This is Nippon bar, who's also aluminum tolerant and has a very similar ART1 protein, and Kasala, which is also aluminum sensitive and have a slight variation of the RX64 allele. And we see that there's very clear differences in the different alleles in their ability to activate beta-gal, although you might expect to see the opposite result, where the sensitive are actually less capable of activating the reporter. So right now we're scratching our heads and trying to explain this result, but also have to remember that this is a single cell heterologous system, and a lot of the other proteins that are part of the complex aren't present there. So we're also moving into a closer system using Arabidopsis protoplasts to try a very, very similar thing. We're going to have a reporter and a promoter or fusion to test different ART1 alleles and see if we confirm the results in yeast. And once we have a system that really mimics what we see in planta, then we want to use this system to also test novel ART1 alleles that we're sequencing from the rice diversity panel. Now, as I said, we're also measuring differences in the different ART1 alleles by just looking at the expression of the downstream genes. And we're doing this primarily using RNA-seq. So imagine that you have the 12 chromosomes of rice, both in your parent RX64 and Azucena, and ART1 here sitting on chromosome 12, interacting with all these different genes in the genome, which we will be able to measure RNA-seq using RNA-seq. And not only that, but we're going to do it in the nils so that we can see how ART1 is interacting with the reciprocal genetic background. Now remember that this is a cross between a Japonica and an Indica, so it will be really important for us to align our RNA-seq to both Japonica and Indica reference genomes. And so we're doing this in collaboration with Cold Spring Harbor, with whom we recently published 3 de novo assemblies of different rice genomes. And we have a PacBio assembly of IR64 that's coming down the pipeline. So we're going to use that to align our RNA-seqs. So we have grown both parents and nils in hydroponics. We have treated them in control in an aluminum solution. We've done this 4 times, so we have 4 biological reps. And the RNA-seq libraries are literally now in queue for the sequencer and BRC. So hopefully in a few weeks we'll have some interesting updates on this. So to quickly summarize what we know about ART1 so far, ART1 is a really interesting example of natural variation that goes across Indica Japonica. And I think the example of ART1 really underscores the complexity of a regulatory mechanism that we are only really able to unravel by looking at natural alleles. And so we think these will help really understand the function and regulation. And in terms of allelic variation, maybe gene expression may play some role, but we're sure that we can measure variation in protein function on the different alleles. And the RNA-seq, as I said, will definitely shed some light in a lot of these unanswered questions. So I want to close talking about this new project that we're part of that was a recently funded NSF Plant Genome Grant that the PI is our own Adam Bogdanov from Plant Pathology. And the grant is QTL editing for crop improvement. And in a nutshell, what we're trying to do is develop a CRISPR-Cas system for genome editing in rice. And this grant has several components that include things like disease resistance, but we also have a small component on abiotic stress. And within that, we have 3 targets that we propose for ART1. We're going to be doing an allele swapping and also a knockout. So we're going to introduce the ART1 coding region from IRX64 into Nippon bar. And we're going to introduce the ART1 from Nippon bar into IR64 and also do a knockout. And you're probably asking yourself why we're using Nippon bar and Azucena, and it's simply because of ease of transformation, and we're really just developing this, and we don't know whether Azucena would be easily transformable or not. Luckily for us, Azucena and Nippon bar have practically the same levels of aluminum tolerance. They're both Japonicas, and they have a very similar ART1 allele. So I think it's a very acceptable replacement. Now, what I think is a bonus feature of this is that we're going to be able to do a proof of concept comparison between the lines that were generated via traditional breeding, which are our reciprocal nils, and compare them with the genome-edited lines. And the genome-edited lines will be looked at in a very similar way. We'll be phenotyping for aluminum tolerance and also doing RNA-seq. And I think it'll be interesting to see whether we have the exact same effect and be able to say, well, we can generate these in 6 months, whereas it took us 4 years to get here. So in the future, I see genome editing as it becomes more high throughput as a way that we can measure, test alleles that will come from RNA-seq and other possible targets of ART1, as well as all candidates that are becoming from the GWAS. Thank you. I want to thank Susan and all of Susan McCuch's lab for all of their support. And I also want to thank our collaborators and co-PIs and tell you that the Rice Diversity Project is mostly funded by NSF Plant Genome, and the aluminum tolerance work is funded by an USDA NIFA grant between myself, Leon, and Susan, and I'll be happy to take questions.